Aloha! I'd like to present our research about quantitative DCE MRI assessment of vascular perfusion in preclinical breast cancer, comparative analysis of the Tofts model and the LRRM model. This is work done at the University of Texas MD Anderson Cancer Center and then was continued at the University of Wisconsin. I don't have financial interests or relationships to declare. So dynamic contrast enhanced, or DCE MRI, has been used to evaluate tumor vascular perfusion in preclinical models for over 2 decades, close to 3 decades now, and has also been used in the clinic, although to a much lesser extent. As we know, normal vasculature is well formed, while the vasculature within tumors is poorly formed, immature, and therefore is leaky. It allows contrast agent to leak out into the tumor and cause the tumor to be bright in a T1 weighted MR image, as shown in the top middle. After giving a drug therapy, you can see that the bottom middle image is then much darker, has a black hole in the middle showing no vasculature, and showing antivascular effects for this particular treatment. However, there's one major technical issue with DCE MRI. There are a number of problems, but there's one major technical issue, and that is that standard DCE MRI requires an arterial input function. It requires the detection or monitoring of the pharmacokinetic uptake and washout within the blood vessel network, known as the arterial input function, that is then inputted into the equation, or the agent is inputted into the tumor by having transport of the contrast agent from the capillary to the tumor. Well, that's a noisy, unreliable signal. I've spent hours of my life hunting for that small dot in the image that represents an AIF. Also, I think it's important to point out that the hematocrit within an artery or a vein that is used as the AIF is not the same as the hematocrit in the capillaries, which feed the tumor. And so there's a systematic error because of this Faraeus effect. A workaround, or a solution, is to then circle the muscle and use that as a reference region tissue for comparison to the tumor. And this model is based on the assumption that capillary networks within the tumor and the muscle are more similar than an artery or a vein, and therefore the wash in and wash out into the tumor can be compared with the wash in and wash out of the muscle, also assuming that the muscle is not affected by the drug therapy if you're looking for using DCMRI to look at early response to therapy. And you can see in the lower left the pharmacokinetic uptake and washout signal for the AIF in red is noisy, and this is actually scaled to be 100% for that, or scaled on this so that you can see all 3 signals on one chart. And while the tumor and muscle are much less noisy. However, there is much less uptake in the muscle, which has an important consideration. Okay, so for the experimental timeline, we made a rather standard 4T1 tumor mouse model, waited until the tumor diameter was 5 to 10 millimeters. Then we performed DCMRI before and then 3 hours after, giving a treatment of Comparative A4-phosphate, a vascular disrupting agent, which has a significant effect on the vascular perfusion. And then we followed up again 24 hours later and then sacrificed the mouse. I'll pause for a moment so you can write down all of the details about the model. Okay, now we'll move on. Okay, the pre-contrast T1 maps were calculated, which is necessary for both the Tofts and the LRRM analyses. It was interesting also to find that the T1 relaxation times for the whole tumor, the tumor core, or the tumor periphery, all decreased after giving therapy, and this was a durable change, indicating that something was happening to the composition of the tumor. We did not follow this up with further histopathology to understand what was happening, because Comparative A4-phosphate is a well-known drug with well-known effects, and that data has been published elsewhere. So we calculated concentration maps of the contrast agent, the gadolinium-based agent, washing into and out of the tumor. And you can see that at baseline, there's basically no contrast agent calculated, and by default, by definition. And then there's a significant amount of uptake that then washes out at the 10 and 25-minute time point. The wash-in rates then were calculated. Now, as an important consideration, the Tofts model calculates K-trans, the transport rate for going from the blood vessel into the tumor, and the LRRM calculates a relative K-trans. That would be the K-trans of tumor divided by the K-trans of muscle. So K-trans and relative K-trans are a bit like comparing apples and oranges, but it's a pretty good comparison, as you can see by these graphs, that both of these methods did see a severe decrease in vascular perfusion 3 hours after treatment, and that rebounded after 24 hours of treatment. Importantly, the T1 data showed that there was still an effect in the tumor at 24 hours, but the vascular perfusion had recovered, as shown by this data. Now, the same result is shown in the tumor core and the tumor periphery. I won't dwell on this. The washout rate, KEP, from the extracellular extravascular compartment, E, to the plasma compartment, P, so KEP, also showed a mild decrease and then a rebound back to baseline for the most part, with both models. And both models did calculate about the comparable results of around 0.1 to 0.15 on the y-axis. So the LRRM performs just as well as the Tofts model. And then the tumor core and rim, tumor periphery and tumor core also performed as expected as the whole tumor. Okay, so if we were to compare the LRRM and the Tofts models, now the Tofts model calculated p-values that were comparable to the LRRM, except from the far right columns show that at minus 24 to plus 24 hours, the LRRM performed better. And this was seen in the tumor core wash-in and tumor periphery wash-in. However, the washout rates were actually more significant when determined from the Tofts model. And we believe that's because the washout rate from the muscle is relatively slow, so that comparative washout rate or washout rate calculated for the tumor relative to the muscle is a little more challenging to do than just calculating the washout rate from the tumor to the AIF when using the Tofts model. So wash-in rate, good for LRRM. Washout rate, better when using Tofts. And the effect size also corroborates this. We see a much better effect size using LRRM for wash-in and better effect size for wash-out when using the Tofts model. So T1 relaxation can see changes in the tumor. DCE MRI, both models work well. Model sensitivity, LRRM is better for measuring wash-in. Tofts is better for measuring wash-out. And the impact of this? Well, I think most people are more interested in the wash-in rate. We don't wait too long, or we don't have a long scan time in the clinic, and so it's better to look at the wash-in rate. And so we really think that the LRRM should be preferred for clinical research, or for clinical trials and clinical studies in the future. So with that, just like to thank everybody and our funding source, and I'd like to thank you for your attention.